yer yer İngilizce.
So I will switch back and forth between English and Turkish, so I would like to apologize for that in advance. Now, the title of our panel is Unmet Needs in Psychopharmacology. So I will be talking about these unmet needs, and this is how I devise my speech. On the one hand, on schizophrenia, there are so many unmet needs, and in bipolar and in depression as well, there are unmet needs. And in schizophrenia, there are new studies, and I will talk about the new perspectives that offer us hope. Also, I'll be talking about how these unmet needs can be met, and also why we fall short of meeting those needs. As you all very well know, the currently existing antipsychotic medications have certain shortcomings and fallbacks. Although they are potent, they were all serendipitous discoveries that were found during other sorts of studies. None of them were rationally designed to treat schizophrenia, I'm sure, as you all know. And when there is serendipity, you have, dopamine receptor blocking as a result. But when we take a step back and look at it, we see that yes, they were found by chance, and from a mechanism point of view, they're not really that rational. Here's an example. You are sitting in a room, the room is too hot. What would you do to get rid of the heat? I mean, it depends on why the room is hot. If there's a fireplace, if there's a flame in the fireplace, that might be the reason, or it might be summer. I mean, if it's cold outside, though, you can open the window, but if it's hot outside, then opening the window will not make sense. Likewise, if we don't know the pathophysiology of schizophrenia, we will not really able to use the medications in an efficient manner. This was discussed 2 days ago during some of the seminars. Hoca Nasreddin says, I lost my keys, and he looks for it in front of the house. People ask, why here? Did you lose it here? He says, no, I lost them in the basement. And people say, why are you looking for your keys here then? And he says, because it's dark in the basement. So, yes, these are quite efficient, effective treatments, but mortality in schizophrenia is markedly high in these patients. So by using these drugs, we don't necessarily save lives. And actually, to the contrary, as years go by, mortality rates are deteriorating. For the last 40 years, the phenomenon of schizophrenia patients dying at a younger age has become much more widespread. I mean, probably this isn't due to the medications. There are other factors that contribute to this outcome. But we can't really say that by using these drugs we do save lives. And medications have shortcomings. So how about novel clinical approaches? What are we trying to do, and what sorts of new treatments can we devise? First of all, we learn more and more about the biological abnormalities of schizophrenia and brain disorders in schizophrenia. First, we need to really know them well in order to devise focused and targeted treatments. And this was discussed yesterday by people coming from pharma companies as well. I mean, subjective reports of the patients, yes, we will take them. Clinical response is important. What the patient says is important. But there needs to be other sorts of measurements, cognitive measurements, physiological measurements, because if we are intervening in the disorder, then the result of our intervention should be determined in ways other than what the patient says. Now, I'm going to talk about 3 new medications. 3 of these, well, I will also talk about others, but I will focus on these for a while. one works on the GABA system. This is a new molecule, and the other on the nicotinic acetylcholine and glutamate. And then I will talk about bitopertin, which was recently developed. As Ali Safet also mentioned 2 days ago, in schizophrenia, glutamate and GABA systems are intertwined, especially paravalvurin secretion that comes from GABA cells, from a physiological point of view, are very important. And in post-mortem studies, we see great disruptions in schizophrenic patients. They are smaller in number, and also the enzymes that synthesize the GABA molecule, the amount is low, the transporter amounts are low, and the protein levels are low. There are also damages in GABA receptors, and these interneurons, these GABA cells, actually organize glutamate cells. They regulate them, and they determine the level of action or activity in the brain. And when also these have an impact on gamma oscillations in the cerebral cortex, and also have an impact on the working memory and cognitive deficit. So the GABA cells are of great importance, and we need to support these GABA cells, and this would improve results for schizophrenic patients. Lewis et al., their study works exclusively on GABA disruptions. These are glutamate cells, and these blue ones are GABA, and this one is paravalvurin cell. And when this synapse occurs, especially GABA A receptors should somehow be reinforced, or agonists should be found, and I think this would bring about great results, because this is part of the physiology that is wrong, that is not functioning well in schizophrenic patients. David Lewis has certain studies, and Merck, the pharma company, developed this molecule MK0777, and probably you haven't heard of it, and there's a reason for that, which I will go into shortly. This was discontinued, but actually it made a lot of sense, so I'd like to tell you a little bit about it. This is a GABA type A receptor. It can be safely given to the humans, and clinical studies were carried out, placebo-controlled schizophrenia studies, and what they did, they used BPRS, brief psychiatric rating scale. This is a scale that was used on the patient, and the patient was assessed, and also patient's performance was also measured by AMBAC, an AX continuous performance test. So the cognitive capabilities of the patient was measured, and they wanted to see whether agonists really worked on the GABA A formulations, and the results were promising in the continuous performance task. In the effect size, the result was 1.07. This is quite substantial. I mean, that this is a serious improvement in the condition of the patient. And also, in another cognitive test, it was 1.74. The patients that took this drug had cognitive improvements. Also, EEG was used to check the brain, and you see that there are serious changes in the brain. In the placebo arm, in the prefrontal cortex, there is no improvement. The ones that took MK0777, there are improvements in the brain waves. So clinical measurements aside, there is a biological improvement as well. But unfortunately, the reason why you did not hear about this molecule is because in the subsequent trials it was revealed that it was not efficacious for schizophrenia. Another group conducted another study, and there were 60 patients, and there was a placebo control, and 2 different doses were tested. In cognitive abilities, there was no improvement, neither in the clinical measurements. So this is a negative study, And yes, the first study was quite promising, but the other subsequent ones were not so promising. So Merck discontinued, abandoned this project. So this is not going to be launched anytime soon. Nicotinic acetylcholine receptors, there are so many things that we know. We've talked about GABA, and we will talk about glutamates as well. But smoking, well, we know that schizophrenic patients smoke a lot, and nicotine actually has an impact on many different processes in the brain, and especially P50 suppression is one of the mechanisms that come into the foreground. This is a bit like sensory filtering. Visually, auditory, other mechanisms are filtered through nicotine, and alpha-7 especially, subunit, alpha-7 subunit nicotine receptors have certain disturbances in schizophrenics. So literature is emerging. Why do patients smoke so much? Because when they smoke, the nicotine that enters the body actually has certain positive effects on the brain. And they sort of correct the disturbances in the acetylcholine receptors in the brain. So why don't we give nicotinic agonists to the patients? Maybe we could duplicate the effect of the smoking in the brain, because smoking also has detrimental effects on health. So there are certain medications that have been developed with this rationale. DMXB-A is one of the molecules. This can also be safely given to the humans, and patient trials were conducted after preclinical trials. Robert Friedman is a psychiatrist, is pioneering this study. And here as well, you don't just ask the patient, how are you feeling, how have you been? You look at the physiological aspects, and you do cognitive tests. And in placebo-controlled trials, it was checked whether the patients who received this molecule exhibited any improvements. And yes, there were serious, significant improvements in P50 inhibition. So instead of smoking, when the patients take nicotine agonists, their sensory, overstimulation or sensory stimulation, filtration is achieved. This is a graphic illustration of what I just showed you. Again, in cognitive tests compared to the placebo arm, there is improvement in DMXB patients, and this is P50 suppression. I will not go into details. However, you see these lines, these vertical lines here, in every P50 tracing, and a downward trend is a negative thing. It shows that there is no filtering. And in placebo patients, there is a lot of downward trends. But when the patients take this medication, it is suppressed. So there is a direct impact on the brain waves in a positive way. In BPRS as well, in clinical measurements, there are advances, improvements. In placebo patients, there is no improvement. But in mild and low dose patients, in symptoms as well, there is improvement. But unfortunately, this was also dropped after the second and third trials. It wasn't deemed promising. It was done by Robert Friedman, and the second study. In cognitive measurements and symptom measurements, they didn't really see a lot of improvement. In negative symptoms, there was a slight improvement. But what they mean by improvement is this. Changing rates is 0. Placebo is shown here. Yes, it is statistically important, but this is the picture, only a couple of points in patients in terms of improvement, which is very difficult to, and it's very difficult to be thrilled about this. That's why the DMXPA was also, it wasn't entirely dropped, but it's progressing really slowly. There are unmet needs, and there are reasons why these needs are not met. There are so many important studies that make a lot of sense, but in the long run, we see once again how complex brain is and how human lives are complex phenomena. So we can't succeed always. Finally, I'd like to talk about glutamate. Nesrin Dilbaz, Safettin, my other professors talk about glutamates. And in schizophrenia, the glutamate system, and especially in NMDA receptors, there are serious disturbances. NMDA antagonists are psychotomimetic. There's postmortem evidence, and in animal models using targeted disruption of NMDA receptor subunits, there are schizophrenia-like outcomes. So it's very difficult to get hold of the NMDA receptor through pharmacology, because if you directly stimulate it, what you can get is seizures, epileptic seizures. If you block it directly, you can cause a coma. So pharmacologically, it's not a very good target. But indirectly, it's possible to get hold of these NMDA receptors, access them. Stimulating metabotropic receptors is one of them. And there is a lot of detailed background on this. Pharmacologists have been working on this. One of the studies focus on mGluR23. This is a receptor, presynaptic receptors on the glutamate cells. And this receptor, if we can somehow block this, glutamate may stimulate NMDA receptors. Lilly developed a molecule, LY 2140-023. This was published in 2007, and this was a very positive study, and people were so excited when this was first published. It was quite a positive study, and this is a double-blind, placebo-controlled, three-armed study. It was quite a serious study, and this new molecule has nothing to do with dopamines. It stimulates the receptors, and it's as effective as olanzapine on the symptoms of schizophrenia, and the side effects had nothing to do with olanzapine. They were like placebo: no weight gain, no sleepiness, no drowsiness, nothing. So it was quite exciting at the time. It looks great, quite effective, and a very low side effect profile. But unfortunately, in the third study, in the subsequent studies, the results weren't so impressive. And Lilly, after the initial positive study, there were two more negative studies, and Lilly had to bury this molecule. They put it in a casket, and they had a funeral. It's never going to be released. LY2 receptors are quite, metabotropic receptors are quite important, and many pharma companies are working on this. Yes, this molecule failed, but hopefully in the future there will be more. And finally, when we're talking about NMDA receptors, we need to also talk about glycine binding. We can't stimulate the NMDA receptor. Maybe coagonism might be used. Glycine and D-serine are coagonists, and they do not cause epilepsy, but still they may increase the function of the NMDA receptor. That's why Roche has been working on bitopertin. This is a new molecule, and second phase 2 studies have revealed certain promising results, and the phase 3 studies are still underway, and we will see what happens in the end. So the first 3 mechanisms failed, and hopefully this fourth mechanism will bring about some good results in the end. We have talked about unmet needs, and finally, here's a couple of things I'd like to leave you with. In schizophrenia, in the last decade, we have seen many developments, and these are upstream. It shows that this is an upstream battle. Like, we set off with dopamine receptors, G protein, matched receptors. We are going into the cells now, and we are trying to get closer to the synapses, glutamate and GABA synapses, because if we can regulate the functionality of the synapses, because in schizophrenia, disruption is not about a lack of chemical substance. It's about a disruption in the activity. We need to change this. We can do this with dopamine, or we can go to the synapse, the source of it all. If we can reach the synapse, if we can change the systems of GABA and glutamates, then we will have a faster onset of action, and we will also have more effective treatments if we can accomplish this. And this is the hope. And Serdar is with us here today, and he also talked about certain studies previously. He's also doing certain studies, and this afternoon as well, I think he's going to be a speaker at a panel. So what he does is this exactly. Changing the activity of the circuits in the brain, trying to reach the glutamate synapse, and devising new treatments, or tailoring some existing treatments for schizophrenia. These are all very promising. And finally, yes, there are promising starts, but we fall short of achieving truly transformative advances. We fall short of doing that for the time being. And yes, there's exciting stuff going on, but because of the complexity of life, we find ourselves in a deadlock. At the lab, we devise ideas, but when we apply them on the patients, the results are not as good as we like them to be. But we continue to learn about the biology of schizophrenia, and one hopes that the treatments will also develop along the way, and we will have improved tools for rapid and targeted drugs in the future. And one final thing that I'd like to say is this. Yes, there are unmet needs, but in psychiatry, because the needs are not met, we train ourselves to be humble. We say, this is what we can do, this is what we can do. But this is not acceptable. In CA, in the last couple of decades, many strides were achieved. I mean, in the past, it was the case with them as well. They used to say, well, our hands are tied, we can't really do much. But now, they're really motivated, and they're doing biological research, and they are making great strides in this regard. And the outcomes are improving. Response rates of the patients are improving in oncology. So I think we need to do the same in psychiatric studies. We need to be more demanding, and we need to demand that we meet the needs of our patients. Thank you.